let's discuss quantitative traits and evolution through space. For this, I'm going to be drawing from a number of examples that involve lemurs because A, lemurs are adorable, and B, with over 50 million years in isolation on the island of Madagascar, lemurs have undergone an impressive adaptive radiation, filling a variety of different habitat types, exhibiting a diversity of social structures, behaviors, and so on. As a result, we can look at lemurs and actually see a lot of these different evolutionary processes at play within this group. Talk about quantitative or polygenic traits. These are traits that vary continuously, and they're often encoded by a very large number of genes. Remember, phenotypes can be continuous, but the underlying genotypes are not. Variation in DNA is discrete. We can identify the genes that are involved using QTL mapping, so looking at those quantitative trait loci. Or we can use genome-wide association studies. Both of these techniques essentially correlate genetic differences like SNPs or single nucleotide polymorphisms. These genetic differences are correlated with phenotypic differences, the trait value, you might say. The body size of ring-tailed lemurs is an example of a quantitative trait, and its trait value is affected by gene-gene and gene-environment interactions. Before we get any further, we need to review a key concept in statistics, which is the normal distribution. The normal, or Gaussian distribution, is bell-shaped and symmetrical around a mean, indicated here as x bar. Variance, then, is related to standard deviation, and both of these essentially describe the degree of variation around that mean. Variance is represented as sigma squared. When variance is low, most values are close to the mean. The curve looks tall and thin. Whereas when variance is high, there are more extreme values, so the curve appears relatively stout, shorter and wider. Quantitative traits can vary because of their underlying genetics or through interactions with the environment. For example, ring-tailed lemurs are normally very similar in size, so this tells us that phenotypic variance due to genetic influence is low. However, malnourished lemurs in degraded, fragmented forests are thin with stunted growth, so the phenotypic variance due to environmental influence is actually very high. The evolution of quantitative traits doesn't require new mutations, just changes in the frequencies of existing alleles. These changes can be brought about through directional selection, stabilizing selection, or disruptive selection. In any case, don't forget that natural selection acts on individuals. Evolution occurs at the population level over multiple generations. As I mentioned from the start, lemurs have been isolated for over 50 million years and have greatly diversified during this time. Directional, stabilizing, and disruptive selection have acted on and continue to act on the many different traits that are exhibited by the 100+ species of lemurs that exist today. Directional selection favors individuals with trait values above or below the population mean. This results in shifting the mean to the left or to the right, but this may or may not affect the phenotypic variance. In this example of fur color, in open, dry forests and at low elevations, lemurs are under positive directional selection for darker hair on their foreheads and around their eyes. Stabilizing selection favors individuals with trait values near the population mean, which would be the optimum phenotype in that particular environment. In the previous case of directional selection, the optimum phenotype was to the right of the mean. But stabilizing selection does not change the mean trait value. What it does is reduce the variance around that mean. In the evolution of lemur fur color, in theory, because lemurs can use color patterns to tell apart different species, communities consisting of more than one species may pressure each lemur to deviate less from that population mean. Finally, we have disruptive selection, in which case it's the extreme phenotypes that are favored. Disruptive selection results in a bimodal distribution. It looks like it has two means or peaks. Ultimately, this type of selection increases phenotypic variance. Now, speciation can be a result of disruptive selection, but probably not in lemurs specifically. Many lemurs are nocturnal and rely on odor or auditory cues rather than visual cues to find each other in the dark. In that case, their fur color might not matter and may not be under selection at all. We can understand the effects of directional, stabilizing, and disruptive selection by comparing the relative fitness of different values of a trait. A fitness function relates some measure of fitness with the value of a trait. Survival and reproduction are common correlates for fitness. In black and white ruffed lemurs, infant survival is higher for mothers exhibiting more social behaviors. The fitness function in this case has a generally positive slope, as communal lemurs have a higher fitness than their solitary conspecifics. Let's take a closer look at a few examples of fitness functions and how these functions indicate whether there is directional, stabilizing, or disruptive selection for that trait. The vomeronasal receptors of mouse lemurs has a fitness function with an increasing slope since there is positive directional selection for enhanced intraspecific communication and detection of predators using pheromones and odors. By the way, directional selection doesn't always produce a linear fitness function like this one. Other generally sloping functions also include asymptotic, saturating, and logistic S-shaped curves. The body size of Verreaux sifaka has a concave unimodal fitness function as there is stabilizing selection for agility and male-male combat. Recall that stabilizing selection does not change the mean value of a trait, but it does reduce the phenotypic variance. Finally, the opsins or photoreceptors of Eulemur lemurs may follow a convex or U-shaped fitness function if there's disruptive selection for luminance versus color contrast. Better luminance, brightness contrast, necessitates only two different photoreceptors, whereas enhanced color vision requires 3 or more. So both of these come at a cost of the other and can produce this divergence we see in a case of disruptive selection. In all of these examples, natural selection creates something that we call a selection gradient. A selection gradient is the strength of selection on a trait, so it can be found along the population mean response, which is the slope beta of the regression line in a fitness function. Note that a selection gradient is analogous to the selection coefficient of a monotypic trait, one that's encoded by alleles at a single locus. But fitness isn't just a function of a single trait. Selection can favor combinations of traits. In n-dimensional space, a selection gradient becomes the adaptive landscape. This means that our two-dimensional regression line from before becomes a 3D or multidimensional plane. The adaptive landscape represents mean fitness as a function of allele frequencies or mean values of 2 or more traits. If the fitnesses of 2 traits are inversely correlated, this is an evolutionary trade-off. For example, there is a genetic correlation between body mass and leg shape in Verreaux sifaka. Genetic correlations may be due to pleiotropic effects, where one allele affects multiple traits, or through linkage disequilibrium, in which case the two alleles are close together on the same chromosome. The evolutionary trade-off in Verreaux sifaka is that an intermediate body size is favored for fighting, chasing, and lunging, while longer, stronger legs are needed for arboreal movement. There may also be additional evolutionary trade-offs since there's little sexual dimorphism and females are not engaging in all of the same body size dependent behaviors as males. Evolutionary trade-offs may be conceptually related to evolutionary constraints. An evolutionary constraint, phylogenetic constraint, or biological constraint is something that prevents adaptation from occurring. A common constraint is a lack of pre-existing genetic variation. We see this in the low metabolism of the red-tailed sportive lemur and other lemurs, which limits the evolution of more active life histories and behaviors, especially during the dry season in Madagascar. Of course, trait change isn't always evidence of an evolutionary change. Phenotypic plasticity is the capability of one genotype to produce multiple phenotypes in different environments. The circadian rhythm of the gray mouse lemur is a great example of phenotypic plasticity, as its internal clock changes from one season to the next and depending on food availability. When we look at the distribution of adaptations on a spatial scale, it's common to find that individuals in local populations are better adapted to their specific habitat than individuals from other populations. Through gene flow, alleles can move from one population to another. Gene flow can occur through dispersal or migration, which is the movement of individuals and/or gametes, genes, across space. Gene flow can also occur through introgression or hybridization, especially in hybrid zones, which are edges or overlap in species ranges. Introgression is actually a type of hybridization in which alleles from one species or population are introduced into the gene pool of another, but rather a small amount instead of a relatively even mix. There is a long history of gene flow among white-headed lemurs and related species or subspecies of brown lemurs. Is this gene flow in Verreaux sifaka? It sure is, or at least it could be, if this individual and this individual's alleles are moving from one population to another. Depending on the spatial distributions of populations, gene flow can be expressed in two different ways. When there are discrete patches, migration rate describes the proportion of individuals in a population that originated from another population per generation. But in a continuous landscape, gene flow is measured by migration variance, which is a measure of the distribution of individual dispersal or migration distances. We're looking at variance, the square of standard deviation around the population average displacement. For the population average, this is theoretically zero, since dispersal or migration can occur in any direction out from that origin. Low migration rate increases inbreeding in collared brown lemurs, but minimizes risks of predation, hunting, and thermoregulatory stress, but that is an eco-evolutionary trade-off. Uncontrolled or overwhelming gene flow into a population can cause genetic pollution. This may also be referred to as genetic swamping, or in some cases, genetic takeover. At high levels of migration, introgression can quickly turn into genetic pollution or genetic swamping. Gene swamping is particularly problematic because it interferes with the process of local adaptation. A fixation index, F sub st, measures differences in the genetic structures of populations, while F sub st ranges from 0 to 1. Because it's generally used to detect genetic differences among populations of the same species, the actual values will almost never be that high, nowhere close to 1. Identical populations or subpopulations have an F sub st value of 0. Populations with small but insignificant differences have an F sub st of 0.05 or less, while those with major differences have an F sub st of 0.15 or greater. Genetic rescue can be used in conservation biology to restore genetic variation in small, isolated populations or those suffering from inbreeding depression. Inbreeding depression is a reduction in fitness resulting from crosses between closely related individuals, and it's often the result of a population bottleneck. This reduces genetic variation and increases the risk of fixation of deleterious alleles. Madagascar is a biodiversity hotspot, but faces unprecedented levels of habitat loss due to changes in land use. Sadly, 90% of lemur populations are currently in decline, and at least as many are threatened by extinction. The gray-headed lemur is critically endangered. Gene flow from related species may help in the conservation of the gray-headed lemur, but the risks include gene swamping, loss of local adaptations, and outbreeding depression. Outbreeding depression is an overall reduction in fitness that results from crossing two genetically distinct groups. Dispersal is expected to play an important role in the ability of species to adapt to global warming and other anthropogenic global change. Several factors can select for dispersal ability, including habitat disturbance, competition among close relatives, and inbreeding. However, species ranges are limited by a number of factors. Dispersal barriers are essentially a loss of interconnected habitat. They are often physical, like mountains, rivers, or roads and farmlands. There are also climatological limitations and ecological constraints as well, including resource availability or the presence of predators, parasites, or pathogens. There are also energetic trade-offs, like the physiological cost of dispersing/migrating or producing a dispersing/migrating phenotype. Okay, next there are genetic constraints, such as the existing genetic variability in a population. There also happens to be increased genetic drift in small, fragmented populations at the edges of a range, along with founder effects and/or inbreeding. Finally, there could be local species that are already present in the area that were there and continued to be well adapted even in a changing climate. So this can limit the immigration of new species into that area. Cockerel sifaka and other lemurs are threatened by ongoing climate change. The International Union for Conservation of Nature, or IUCN, consider lemurs to be the most endangered group of vertebrates on the planet. Climate change poses many challenges for lemurs. One effect includes changes in tree phenology that alter the availability of fruit, which is a keystone resource for lemurs. Another effect is the increased frequency of droughts, which is stressful and prevents at-risk lemur populations from recovering. All of these effects are exacerbated by human disturbance. However, there is still time to make a difference, and we can apply studies of ecology and evolution to aid in lemur conservation. Thank you for taking the time to watch this video. If you have any questions, feel free to reach out to me.